Hello everyone, how are you guys? I hope this is a great day for all of you. So with me, I'm Raihan Mediotama with number ID 13086200032. In this video, I want to talk about RNA structure, the structure of that single copy of the genes that codes for a protein. The RNA nucleotides looks like DNA nucleotides, but there are a few very important differences. So the RNA nucleotides also has a phosphate. Here is one of the difference. It has having ribose sugar instead of deoxyribose sugar. So RNA is ribonucleic acid and DNA is deoxyribonucleic acid. And the third component is nitrogenous base. It's like in DNA, it has the same exact nitrogenous base on its three-part nucleotide. So in general, a nucleotide is a sugar, a phosphate, and a base. The difference is depending on nucleotides for the sugar change. So think about ATP. ATP is nucleic acid. It is made up of nucleotides. The difference here is that ATP has three phosphates, a ribose sugar, and a base to the side. And then RNA has a phosphate, a ribose sugar, and a nitrogenous base to the right. The DNA has one phosphate, a deoxyribose sugar, and a base to the right. So the nucleotide structure is always a phosphate, a sugar, and a base. One big difference in RNA is its structure and the types of nitrogenous base that RNA has. So one of the first one is the same adenine, except adenine has a different partner in RNA than it does in DNA. Adenine, instead of pairing with thymine, is going to pair with molecule called uracil. So uracil very similar to thymine, but it look at a different chemical component. So it's different entirely. So adenine with uracil in RNA and in DNA, adenine with thymine. The other two are exactly the same. Guanine always pairs with cytosine. So the only difference is nitrogen basis of RNA is that adenine with uracil. The RNA molecule does have some differences from the DNA. First is that RNA is only single-stranded. So remember, we said that DNA can never leave the nucleus because it's too big. Well, part of the reason that RNA is single-stranded is so it can leave the nucleus. Next, RNA is made by copying a segment of a single stranded of DNA, so it is not the entire genetic code, it's just a piece of it. Third, it uses uracil instead of thymine bound with adenine. And last, it leaves the nucleus to go to the right ribosomes in the cytoplasm. So to tell you if it's going to arrive soon and must be used in making proteins. Now, there are a few different types of RNA that become very important when in making proteins. So the first type of RNA is called messenger RNA. The second type of RNA is ribosomal RNA. And the third type of RNA is transverse RNA. First type of RNA is called messenger RNA. Messenger RNA is a copy of the gene sequence form one strand of a DNA molecule. When it is complete, it leaves the nucleus and goes to ribosome in the cytoplasm on the rough endoplasmic reticulum. The second type of RNA is ribosomal RNA. It is a molecule that makes up ribosomes, makes up the proteins building unit, and ribosomal RNA helps to make proteins during process called translation that we'll get into a little bit later. Third type of RNA is a transverse RNA. Transverse RNA helps to transfer amino acid to ribosomes when they are needed. So that's it from my presentation in this moment. I hope you can understand what I'm talking about, and thank you so much for watching. See you.